So I'll go ahead and get started. So first up today, I'm going to tell you kind of a story about this project we've had in my lab for a long time. Um, and it's kind of evolved, and the pandemic actually forced its evolution in many ways. And I'm gonna tell you a little bit more about that today. So first up, let's let me show you a map. All right. So for those of you that don't know, the Great Salt Lake is a extreme hypersaline environment in Utah. I'm in Idaho, so it's literally down the street from me. And so it's been a very popular place for me to take my students because it's a great example of an extreme environment, an extreme hypersaline environment, and for showing halophiles in the wild. Because my students see a lot of wildlife here in Idaho. They don't realize that microbes can be wild too. And I know all of us will appreciate that. It's when you teach students, it's always a new thing. So so on that note, well, we go down the street, we can go there, and we usually sample in the spiral jetty. And I'll kind of use this to show you. That's the spiral jetty right there. Now, when some things to know about the lake is this lake is bisected by a railroad bridge. When they made this bridge back in the 1920s, it was done by something called a mound strategy. And this is important because this means they mounded earth across the lake and effectively cut off circulation of the water between the two halves. This effectively caused the northern end to be hypersaline, and you can see this in this very beautiful photograph, actually from the ISIS. I know we were talking about that before, we got started here. Anyway, it effectively cut off the concentration, and the halophiles bloom in the northern end. And I don't want to give it away too much, but the halophiles also bloom in the southern end, and we'll come back to that. But I had this problem. I couldn't sample anywhere other than the spiral jetty, mostly because the rest of the land around here is private. And to get sampling privileges, it's difficult. We'll just say there's a lot of paperwork. So because of the pandemic, one of my colleagues in geology was retiring. I was telling him, well, kvetching really, about being unable to sample in anywhere else. And so he actually put me in touch with the Utah Geological Survey, and they actually drove me out in June, so during the pandemic, to go sample on the causeway. All right, which is a pristine environment. So this kind of goes to one of your agenda items, Scott. There's no people here except a railroad. And I have a lot of funny stories about the railroad. I won't waste our time on that. But if you ever want to hear a funny story, I got chased by a train. That's all I'll tell you. And we'll go on from there. So anyway, I could sample across this bridge. So I actually got a sample in the northern part, and I got a sample in the southern part of the lake, and decided, hey, what a great time to learn more on bioinformatics. So you'll see how that worked out. All right. To kind of, I feel like I need to show you guys how we sampled and what we did with the water. So a couple of things. Because I'm in a chemistry department, I have a lot of environmental chemists as my colleagues, and they knew I was doing this metagenomics on the water. They also wanted to look at the metal content of the Great Salt Lake, because it's been characterized many times, but the lake is in a drought and concentrations of metals are going up. And of course, this means the halophiles are getting more intense, the metals are getting more intense, and then there's this whole thing about methyl mercury. But that again is another story, but we'll probably circle back to it. So we collect the water. We do a double filtration system, which most people I know do. We have a crude filter to get the junk out, and the eukaryotes. And then we have a fine filter to collect the microbes. And in addition to that, we are also treating water with nitric acid so that we can analyze the metal content, right? By the way, on the metagenomic side, nothing totally spectacular here. We do, I think, what everybody does. We beat those filters, some extractions, a lot of 70% ethanol washes to get rid of salt. We also do a very crazy PCR inhibitor cleanup, because this environment, we'll just say getting PCR to work after this was really difficult. So the cleanup was really, really necessary. And then we could move forward. And then just to also point out, we're actually using V4, V4.5, and V9 primers. So we're doing not only an archaeal bacterial characterization, but also a eukaryotic one. And that's going to become important in a minute. So I promise I'll tell you more. All right, so the first curious thing was, how extreme are we talking? And so I'm showing you a little bit of preliminary data here to give you an idea about the metal content of this lake. First up, it's crazy extreme, as we all pretty much knew, so that's not really new information. The other thing not new is it's a lot of sodium. But the other informative thing we found out was that it's got almost all of group 1 elements represented, most of group 2, and a lot of other metals. I decided I'm not going to show you all of our metal data. We have a ton of metal data here, and it's actually both fascinating and a little terrifying. And the terrifying aspect comes, and this is why there may be a policy shift in this lake, is that some of these metals are not human friendly, and the spiral jetty is definitely a public place, and people go in the water. And I'm actually trying to get them to put up a sign to keep people out, not just from the microbe level, but because of metal. So that was kind of one thing we found out right away. Another thing that you can't see very well, because I had to plot this as a log scale because of the concentrations, is that the concentration of metals in the southern half of the lake are about half as much as the northern. This was a surprise because we thought the north was more extreme. So we were anticipating a bigger difference, but it's not there. So that was interesting. So that was very, very interesting piece of data that came out of all of this.
One quick question was really cool. How would this compare, say, to other, this is within within this lake, how does it compare to like other lakes or other places we see some of these levels, I guess, to context?
Oh, I don't have an answer for you yet. That's actually something that we're figuring out right now. It's the concentrations of certain metals is a lot higher. So one unique thing I can point out right now, just to show you this, and I'll tell you, this is what got my geology colleagues very excited, was lithium. Not many of the lakes have such a high concentration of lithium. But other than that, I don't have many other answers for you. We're still trying to characterize it. Like I said, this is all preliminary, so we're trying to figure out exactly how to present this information.
I will say that the lithium content and some of the metal contents that I'm seeing are kind of consistent with the Great Salt Lake Desert as well. I've done a lot of geochemical analysis in the Great Salt Lake Desert, and the numbers look similar.
Interesting.
Especially the lithium numbers. Good. Pretty similar.
Woohoo! My colleagues will be happy about that. Yeah. I'm glad to hear that.
Yeah.
And how far is your site from the Great Salt Lake?
Oh, my site is next door. It's about for where she at from the causeway, which I've sampled at before too. It's about
like over here.
It probably takes about an hour and 45 minutes, just because you have to basically drive through this bombing field that belongs to the military, and then get back on the highway, and then it's like another hour and a half to basically the Bonneville Salt Flats from there. Coming from Spiral Jetty around, it's more like a 4-hour drive because you have to go all the way around the lake. So it's not, I mean, basically, as the crows flies, it's not very far. I mean, they're all part of the same system. They're all part of Lake Bonneville. They're all connected anyway. So the numbers make sense totally because they're a connected system.
Yeah. It's an insane place. I know what she's talking about too. We had an episode with the bombing range. Actually, that's again another story we could talk about.
I would love it. I'm excited to see this.
Yeah,
data,
you know,
so
right?
More for sure.
Yeah, it was insane. They started testing bombs while we were collecting one year, and it was almost we're like, oh my God, what's going on? Why is there a mushroom cloud over there? So yeah, talk about surprising things.
Yeah.
So anyway, yeah, it's quite the environment, actually.
Yeah, it's pretty crazy.
It is. So yeah, I'll tell my colleague, we were like worried about the numbers, to be honest with you. But anyway, hopefully in the very near future, we'll actually be publishing this. So we'll hopefully get good comments. Anyway, moving on, moving on. So we got this interesting metal data, but in the meantime, we decided to up our bioinformatics game, and explore different ways of characterizing the population. So originally we were using mother, all right, and OTU identification, and that was great. And my students could master mother pretty easily. And but as you may or may not know, mother doesn't resolve, resolves up to the family level, and not much beyond. And we had a lot, so we were getting halophiles, but we wanted to know what halophiles are there, and we couldn't figure that out. So, and then we had a pandemic. And that pandemic convinced me, who was on sabbatical and had all my sabbatical arrangements canceled. I decided, you know what, I will learn how to use the Data2 package and do ASV analysis instead. And that was I have to admit, it's been a journey. I've had to relearn programming, so that was a lot of fun. But the good news is, made a lot of progress, and have some interesting things to share with you. So I'm going to warn you again, these are all preliminary results. I'm still working on the data set, but I told Scott I'd show you guys some stuff that we have found for your information. All right, so I got ggplot to pull this this out. This is the V4 results, so the 16S. And what to show you a couple of things, just really quickly. What you can see, it will surprise no one that the predominant species are halophiles. In particular, the archaeal halobacteriota. So 40 to 50%. All right. The other thing that's interesting, I actually separated them based on filter. So I've got the fine filter and I got the crude filters on here. And I'll be honest with you, I almost threw out those crude filters. And then I said, you know, there's a lot of stuff in there. We should do that. So we did it. And I'm glad we did, because it's actually rather interesting. So I was expecting most of the bacteria to go flying through that crude filter, but we're actually getting some retained. I have a hypothesis about this, so don't worry, there's more to come. Now, in the southern end, the one that's half as concentrated as the northern end, in the fine filter, again, a lot of halobacteria, but you'll see in the crude filter, not so much. We instead have a lot of proteobacteria, and those are legit proteobacteria because I decontaminated my results. So, you know, that's, by the way, if you ever want to talk about decontamination, I can tell you a lot about that. Anyway, so the end result is sort of like this. So halobacteria, halotolerant species dominate both north and the south end of the southern part of the lake, which was a bit of a surprise. We didn't think the halophiles would be as represented. They were. Also, halobacteria are in both the crude and the fine filter. So why might that be? So now to show you the 18S results, and that, I think it dovetails really nice with the 16S. So first up, when you do 18S analysis, a couple of things to point out. V9 primers are terrible for characterizing eukaryotes because you get a lot of contamination. And I know if anyone who's ever done V9 primers, they'll totally be like, yeah, you get a lot. So I had to filter the results for just the eukaryotes. And I got an interesting story. So what you can really see here is that the predominant species here are the, hold on, let me get this up. Come on, there we go. Are chlorophyta, which is this Vanilla species. I'll talk more about them in a minute. And arthropoda. Now, most of us know arthropods, you know, insects. Well, these are the Anostraca. If you look at the species of eukaryotes that are dominating, it's these two. So chlorophyta is a single cellular hypersaline algae. So no real surprise there. They're found in almost every hypersaline environment I've actually seen a paper on. So kind of cool. The Anostraca are really cool. So these are your fairy shrimp. And I was like, really? Now for us oldsters out there, these are sea monkeys. You remember sea monkeys? These are sea monkeys. And here's the really cool part. They eat halobacteria and the Danella plus other algae. And that's, I think, why in the crude filters, we're getting a lot of halobacteria and Danella kind of stuck in those crude filters. They should really be going through. So anyway, that was just kind of an interesting thing we kind of found with these results. Like I said, these are still a bit preliminary. We're still trying to characterize everything, but you can see it's really heavily weighted in some places, like here in the southern end, there's those berry shrimp there. And then on this end, there's that chlorophyta just totally dominating, right? But the nice thing also is that you can see the north and the spiral jetty are very similar. By the way, that was true for the 16S results. And the south is kind of its own thing, but still very halophilic. All right, so that's kind of the big, that's what I got so far on my results. We're still trying to work out all the arrangements, and I didn't want to show you guys diversity plots because they're really skewed. Because we have so many halobacteria, they really skew the diversity plots. I'm working on that. I got to figure out how to do that. So other things to kind of come for the future. So without further ado, some acknowledgements. You don't do anything this stuff in a vacuum. So I have to thank Utah Geological Survey quite a bit. They were awesome, even during a pandemic. My core facility did all the MySeq stuff. Dr. Rosentrader and IAS Environmental Chem did the metal analysis work. Mike Lee has been a godsend for the bioinformatics. He was pretty much able to hold my hand while I tried to learn R and also all those packages. So he's been wonderful. And I've had two really great students who've also helped with the project. Talia Calhoun, who's now at the University of Utah, she's a graduate student, graduated during a pandemic, felt bad about that. And then Alex Guerrero, who's still in my lab, who's actually on another project because, well, we'll just say he has to learn more R. Anyway, yeah, just saying. And then lastly, I can't end a halophile talk without putting up a far end cartoon. The far side, sorry, cartoon with the Great Salt Lake and the slugs, of course. And then I have two shameless plugs. I'm trying to do a Fulbright Award. So if anyone's looking for someone to hang out in their lab, just let me know. And then also, I'm also editing an issue of Life. And if you're interested in contributing to it, please let me know. All right, I'm good.
Thank you, Caryn. Anybody open it up for questions if people have questions and comments or something to add to this?
Hey, Caryn, I have a sort of a suggestion for the diversity metrics. Have you tried to do because we had to deal with the same issue in the Great Salt Lake Desert. What I ended up doing was, did you do a subsample to make, basically for doing the alpha and beta, did you subsample from your population and then do kind of do your diversity metrics from there? Because that kind of helps kind of filter out the overabundance of like the halophiles. If you subsample your population and get an even number and then do your diversity metrics, it kind of helps a little bit.
Oh, I will definitely take that suggestion.
That's what I had to do for mine, because yeah, and that was the recommendation I got. And basically what I ended up doing was subsampling to like whatever my smallest genome, because I had multiple samples, I was doing a transect, so I subsampled to the smallest transect for all of mine to get evenness, and it really kind of helped kind of lay out the diversity metrics a little bit. Oh,
I will take that suggestion. No, we're just combating that right now. So thank you very much. I'll take it. I'll take anything.
Absolutely. And I would definitely love to talk to you one-on-one. You and I should definitely talk one-on-one. I think there's some fun stuff that we could collaborate on.
Oh, I think so too. Yeah. I agree. All right, fantastic. All right, more
Thanks for
questions.
the great talk. Great talk. This is exciting. I love the Great Salt Lake, so I can't help it. I'm a biased fan.
Oh yeah, it's so much. Oh yeah. Anyway, yeah, we can talk. There's so much. All right.
So moving on, any other questions?
I would like to ask if you looked into deeper taxonomic resolution than on the level that you showed.
Oh, so
especially
the answer
for the halobacteria.
Mm-hmm. Yeah, so yes, the answer is yes. What I just showed was what I could cobble together for today. Actually, I'm getting all the way down in some parts to genus level, and it's really been quite informative. In fact, if you believe it, sometimes I'm getting some species information as well. I see there's a caveat in data 2 when you're going down that low. And I'm also working to resolve that. That Mike Lee is really helping me with that. And we're kind of hoping to be able to tell some species information because we can get it to a certain level. And yeah, so we can subspecies the halophiles out as well as the other organisms as well. So we're trying. There we go.
I got a question, Caryn. Have you thought about just doing low coverage shotgun on this?
Okay, so others have done that. And the good news is our results actually match those. We've got more detail. So some organisms that did not pop up in the shotgun are popping up here. So I think that's really interesting. Plus, I think the 18S analysis is actually kind of cool. So I think it's all kind of coming together. And like I said, we're still working out some of the details. But yeah, it works.
One of the things we did on, as you know, Caryn, on Lake Hillier, we did a ton of microscopy, and we actually enumerated the Denaliella species that were in there, and we actually enumerated them in the shotgun too. So we kind of got rid of some of the PCR bias. So it's certainly worth it. Is there DNA left over from your samples?
Oh, I knew you were going to ask that.
I was curious for the same thing. I'm wondering. Yeah, we'll sequence it. Happy to contribute to the study.
Oh yeah, so I have leftover filters in the freezer. So the answer is yes. We can prep more.
Cool.
I made my students make multiple filters. It was yeah. Anyway, it's if you know students, you know how you have to crank the whip sometimes. Anyway.
I had another question. Have you guys had a chance to compare your results against the microbial work that was done by the MSU group, looking at the microbialites? I would be very curious. One of my first questions is, how does the lake water compare to what they sampled, what they did with the microbialite populations that they did in the North and the South Arm?
So your question's very timely. I literally have that open on my desk. I'm actually still trying to match up everything. So stay tuned.
Oh, this is so exciting. Yeah, yeah, we got to talk some more. I just sent you my email.
Yeah, we got to talk. I agree. We got to talk more. All right, cool.
Any other questions for Caryn before we move on?
Okay. You can always email me too, by the way.
Yep, she's on the group.
Okay, hello everybody. First of all, I would like to say hello, good evening to everybody. This is Felipe Gomez from Astrobiology Center in Madrid, and if you don't mind, sometimes I have some problems with the bandwidth. Then I am going to switch my video off until later during the questions, if you don't mind. When Scott told me about the work that we were doing during the May's project, and he asked me to present this paper, I think was interesting. As he said, I am involved in different things. Right now, I am doing very weird things with perseverance, but at that moment, this is a past work that we developed some years ago. And I think it's interesting because it was a really productive work, this project. This paper is one of the several ones that were delivered. I'm going to talk a little bit about the taxonomic and functional analysis of intact microbial communities thriving in extreme astrobiological relevant anoxic sites. And I have to say that this work was not coordinated by me. It was coordinated by Christine Motz-Stenger. And but this paper is not possible to be explained if we don't speak before about the MACE project, Mars Analog for Space Exploration project. It's a really interesting project because it was a collaborative enterprise in order to study different Mars analogs environments from the microbiological point of view. And in order to understand the papers, we need to talk about this project because it's important to figure out why the different sites that we were sampling on were chosen, and at the same time the final result, as I said before, it was really productive project. It was really a work done by different expertise people. This was very interdisciplinary and was really productive, I said. MACE project was supported by European Commission, FP7 project, and it was run from 2014 to 2017. And but as you saw on the paper that Scott told me to present, we are still delivering some papers, writing and publishing some papers, because it was really productive. It was very interesting project. We had 11 partners from the University of Edinburgh, which was the coordinator, Charles Cockell at the UK Centre for Astrobiology. The project officer was run by the ESF, the European Science Foundation, but we already have on board the GLR in Germany, Autonoma University in Madrid in Spain, Matis Institute in Iceland, the Leiden University in Netherlands, and ourselves, INTACAP in Spain, the Astrology Center. This is me, basically, for this project, and the IANA in France, European Astrobiology Network Association, the University of Graz in Austria, CNRS in France, British Antarctic Survey in UK, and the European Science Foundation that was running the project officer. And while looking at anaerobes, the reason why we choose this very particular part of the niches that we were studying was because previous to this project, basically the attempts to investigate the response of organisms in Martian was focused on anaerobic conditions. Surprisingly, most of the work previously done to this project was focused on aerobic. Very few were conducted and focused on anaerobic, and at the same time generally focus on strains from culture collection, not from natural sites, extreme environments. For us was like very interesting to see that there was a niche to research in which we thought to present the anaerobic niches, the anaerobic part of the several extreme environments. At the same time, I have to say that microorganisms under Martian studies normally are usually characterized under very limited set of conditions. This means that when somebody study some microbes to develop on the surface on Mars, normally the study is focused on, for example, the low pressure or low temperature, but not at all at the same time. Then there was a lack of data on combined streams for Mars. At the same time, we thought as well that several stresses might interact. For example, growth on low temperature could affect the water activity range of growth, or salt concentration and high ionization radiation could play some parallel effects over microbes. This is the reason why we chose this anaerobic part of the extreme environments. Of course, mimicking or doing some similarities with the possibility of habitability on Mars. At the same time, we include biosignatures. For example, we were studying the possibility of any kind of signs of life to be preserved on Mars and the stress and conditions of the surface of the red planet. And for example, we were interested in metabolic activity, any kind of fossilized, in this case was CNRS Institute with Frances Westall and Frederica Joubert working on fossilized remains cells of communities. Several aspects that were interested for a final plan in order to work on maize work plan. The idea was collecting new samples from analog sites to try to isolate, growth, and characterize novel organisms, to expose these novel organisms to different extreme conditions, and to follow the production of biosignatures and fossilization of these biosignatures. As I said before, it was a really complex project. And of course, this is the reason why some of the papers are very interdisciplinary. For example, in this paper that we are talking about that today, if you go to the discussion section, you will see that there was a discussion including other works published on other papers, because the final results, the final outcome was really complex. The sample sites that we decide to sample in was mainly 3 dedicated campaigns. Bulby Mine in the UK is a mine 1 kilometer deep in under the North Sea. In a salt, in a very salty environment. In Iceland, the Granavan Lake, which was an acidic lake, and in Germany, the Siebenhauer Moor, which was a sulfidic sulfatara on this site. And at the same time, we got some samples from permafrost in Russia and Rio Tinto. The project itself was not sampling on these 2 sites, but we provide samples from Rio Tinto and from permafrost in Russia. The idea was just use different kind of environments, different kind of parameters at the same time in a natural environment and in a natural microbial isolates. The idea was to study anoxia, low temperature, high temperature, aridity, high salinity, acidity, and low nutrient availability. And on the left column, you can see the different sites. Rio Tinto, for example, we were sampling on sediments and on surface of the origin, the Rio Tinto area. The cold sulfidic springs on the Siebenhauer Moor in Germany, the volcanic environment on Iceland, and the Bulby Salt Mine and the permafrost. All of them, as I said before, was anoxia, were anaerobic analogs, anaerobic environments, but some of them were at the same time low temperature. For example, in the case of volcanic environment on Iceland was at the same time high temperature. Aridity, for example, in Bulby Mine was possible to have very low water concentration, acidity, and low nutrient availability in all of them. This is the reason why we decide to choose these anaerobic environments. DLR was coordinating the artificial fossilization and aging. We selected isolates and were exposed to stresses that were representative from Martian environment. I mean, several parameters at the same time. And we paid particular attention to the combined stresses. And as I said, was DLR the coordinator of this part. Artificial fossilization was studied by CNRS, the lab coordinated by Frances Westall. And these colleagues were studying the agent of these samples that were fossilized, and they ran artificial fossilization of these samples. At the same time, we used some very automated technology for light detection. This part was coordinated by CAP, by Astrobiology Center. For example, we were following life and biosignature detections, protocols and techniques used during the campaigns, during the stress simulation and fossilization in order to try to identify signatures preserved and at the same time any possibility, any study or the detection of these biomarkers, these biosignatures. This part of the work is not presented here in this talk. Okay. And now going directly to the paper, these were the samples. This is the reason why I decided to present a MACE project, because I think it's necessary to understand MACE in order to understand the sample sites choose for this particular paper. We use for this paper, we use the sulfidic springs in Germany, the hypersaline environment in Bulbe mine, the acidic lake in Iceland, Rio Tinto, it's an acidic river in Spain, the permafrost, and at the same time we got some, by coincidence, because it was not expected, to additional samples for a glacier in Austria, with these different characteristics that I presented before. This slide represents the archaeal and bacterial profiles of the intact microbial communities from the different sites, the different samples. The panel shows the taxonomic profile obtained using the archaea targeting primer, or in the case of film, in the upper chart, and the family level below. File names are followed by the relative abundances, I have to say. And at the same time, for the following panels, I have to say that the relative abundances of each taxon is shown on the y-axis. The total relative abundances summed up for all samples is given in brackets behind the taxon names in the legend. Okay. I will summarize later on. I would like to present several panels before. In this panel, for example, shows the bacterial composition of the samples on class level. But just only we choose, in this particular case, we choose the only those classes with higher percentage, higher than 0.1 percentage. Okay. And the final panel here, we summarize the most abundant phyla of the microbial community based on universal primer sets microbiome. Some of tips that I can give to you, it's that if 99% of the sequence detected in the acidic lakes, SS1, this is from Iceland, remain unclassified within the archaeal domain, and thus might represent a new taxon, whereas all sequence of the second sampling site of the acidic lake, SS3, could be assigned only a film level or were classified of potential methane-producing bacteria, for example. All the sequence potentially, for example, as well was identified potentially ammonia oxidizing archaea, eukaryotic, minor proportion of Gusarchiota. I have to say that the sequence from Rio Tinto, for example, were mainly assigned to unclassified archaea. This was practically 72%. Taumarchaeota 10% and Euroarchaeota 16% in one of the sample stations on the river, and the high proportion of eukaryoarchaeota. Just as a summary, I can say that in the spring biotopes, a remarkable high proportion of signatures assigned to Gusarchiota was detected, practically 26% and 21% for all sequence counts in both samples in this environment. In addition, archaeals was already identified. And notably, for example, only the sulfidic springs biotope revealed signatures of archaea belonging to the group of Enermarchaeota. In the case of the permafrost, just on the central part of the graph, was possible to identify mainly signatures of Eurarchaeota with high proportion of methanogenic archaea and other related groups. The microbiome of maize sites harbor a vast biodiversity of adapted anaerobic microorganisms. Intact microbial communities in maize environments were generally characterized by proteobacteria, for example. 27% in this graph can be shown, followed by actinobacteria, Bacteroidetes, Chloroflexi, Acidobacteria, Firmicutes. Some signatures of proteobacteria were present through all sampling sites, but in different proportions. Similarly, signatures of actinobacteria were detected in each environment, with the exception of the hypersaline environment. This biotope also revealed a minor proportion of Firmicutes signatures, which were completely absent in samples from the acidic lake environment. In this slide, we saw the microbial biodiversity and the network analysis of microbial communities in sample from all the sites. In the left panel, it's possible to see, gives some information on the general microbiome universal dataset, whereas the right panel displays results from the archaeoma analysis. Archaea target approach, the alpha diversity, for example, beta diversity, and different kind of parameters in order to identify different the samples from the sulfidic springs. Also, for example, so with highest diversity when observing all microbial signatures. The lowest proportion of some of the intact cells were detected in samples from permafrost, for example. The microbial communities derived from the acidic environments, such as, for example, the acidic river, Rio Tinto, or the acidic lake in Iceland, although obtained from geographically remote sampling sites in Iceland and Spain, were very close, were clustered in both the PCO plots. Some remarkable information that was really interesting and surprising for us. And finally, this slide that I can see that is really difficult to read. You have to trust me. But basically, in this slide, we saw with metagenomic-based functional profiles of selected sites, the sulfidic springs, which is difficult to see. Sorry. Another was the acidic lake in Iceland and the permafrost. We can see here in the first panel the overview of the found functional pathways and functions of and their relative abundances. In the left bottom panel, the differences in the functional profiles of the different sites. In the upper right, the comparison with other non-extreme but comparable sites, for example, soil, groundwater, that not reveal significant impact on the metagenomic profile. And finally, in the right column on the bottom, the summary of the maize sites that differ significantly from other moderate, not extreme environments. Some tips that we can extract from this slide is that the most abundant enzymes function detected in the shotgun metagenomics datasets were an integrase involved in phase integration of an excision. Some glycosyl transfer access, for example, related with biosynthesis of galactoglycans and related lipopolysaccharides, some cation efflux system proteins, long chain fatty acids, decarboxylates, really complex, really interesting information. The methanogenomic derived microbial functions of the three environments showed really differences in the levels of certain metabolic pathways and function. In particular, for example, something to remark is the permafrost sample that was characterized by an increased turnover of amino acids. For example, increased cold shock responses, iron acquisition and metabolism, stress responses. Tremendous information that was published in this paper, very complex paper, but really, really interesting. I have to say that most of the paper that we delivered along the project were very similar. For example, some aged fossilization simulation from some of these isolates were published on scientific report, and at the same time was really interesting. Basically, just for summarizing, I think I am completely out of time. The outcome, I can summarize, conclude that we got a better understanding of habitability and light detection. We developed some light detection technology. We study analogs environments for space exploration. We follow biosignature of life and how to detect them. I think we got an understanding of the adaptation of life, and we studied some limits of life to apply on Mars. And I have to say, I started my talk telling that it was really productive. I have to say that three years later of this project to be finished, paper still are coming out. I think it's one of this interdisciplinary approach in which different people, different expertise, as for example, we are doing here in this group that we are discussing tonight, is really productive, and I think it's something that is really interesting in order to recommend people among us to collaborate and to try to understand in a very particular focus one of the sites or one of the issues. I think this kind of collaboration are really productive in order to provide really really interesting information to be published. And that's all.
Felipe, thank you very much. I appreciate that. It's always interesting to hear what you got your hands into these days. I'm intrigued that there's people doing some stuff with anaerobes. I think they often get the short end of the stick when it comes to culturing these days, at least from what I see. People are happy to go after the aerobes, and that's really not how, probably not the.
I have to say that because I insist that the project was really, really complex. I have to say that we run isolation and growing at the same time. We have a lot of different isolates from the different sites. It was really, I insist, was really interesting project, the maize project.
Do you grow on different substrates like methanol or in types of environments? Or do you modify your media for what you're after?
Yeah, for example, one of the most study areas, and this was the reason why we decide not to sample was Rio Tinto. Rio Tinto, we had really well studied this environment, this acidic river, and we have a lot of different isolates, including methanogenic bacteria, and at very low pH. I had to say that. And we have growth these isolates in different kind of mediums, from fatty acids, not fatty acids, mixing cultures with acidogenic. It's really complex. And we did similar studies with, for example, permafrost, but in the case of permafrost, we got limited access to the permafrost, then the study was more limited.
Any questions?
Just one, like I was seeing you have done a very nice analysis, Philip. Where can I access the codes for it? I couldn't find a GitHub link.
Sorry?
I couldn't find a GitHub link for the analysis that you did.
Okay. Okay, I can check for you if you want.
Yeah, sure.
It should be available. I have to say that finally this paper was published recently. Probably it's a problem of time but I will check for you.
Sure, thank you.
And some others remains to be published, I have to say.
So one of the things that we noticed with acidic lakes in the past is getting DNA or RNA from those samples can be challenging, really challenging. And I was wondering what kind of steps you took to get the sample ready for extraction so you could actually get a reasonable amount of DNA.
Normally, for example, we have the advantage of being working for several years in Rio Tinto. Rio Tinto is a really interesting environment. Probably in some future discussion, I can present Rio Tinto for all of you. It's really interesting because it's very acidic river, and we have been working there for probably during the last 20 years. And I think this is really is an advantage, because it's really difficult to extract some organics, or for example DNA of overall RNA from these samples. Normally we choose several we follow some protocols and are standards, some different kits in order and normally we use chelators in order to sequestrate all the metals and to complex the DNA in order to avoid the degradation before the final steps. For example, with acidic water. At the same time, I have to say that, for example, in Rio Tinto, some of the stations are really interesting because there is very important patterns of fossilization, metals deposits, and layering, in which the microbes are encapsulated very fast. It seems like a rock, but when you study the rock, you discover that it's not a rock, it's a different layers, precipitated, and inside you got different kind of bacteria that are stable, but of course when you modify these conditions, normally the organic matter is very fast degraded. This means that you have to act in very fast way in order to avoid this precipitation, and we follow different protocols in order to chelate metals and to avoid the acidic water.
Yeah, I could tell you we have a pure strain from Shaban, who's also on the call right now, from Romania. And that's an acidothiobacillus. And we've just started working on this thing, but getting large amounts of DNA has been somewhat challenging. So I was just wondering if you had, and you can respond to me offline or to the whole group, but anything to get DNA from acidophiles would be appreciated to see what you guys have done, if you can share.
Yeah, of course. And I have to say that normally the cell counts, the cell numbers are really low in these so extreme environments. It's really low. Then sometimes it's really difficult to isolate, and at the same time to have an enough quantity for later process.
It's DNA, really challenging. Based on the number of cells multiplied by 3 femtograms per cell, we're not getting a calculated amount of DNA that we expect to get out with at least a 10% efficiency. So that's one of the
advantage of this kind of very acidic environments is, for example, in our case, we were studying the effect of UV on organisms, for example, on bacteria, and we had the possibility of having high concentration of different metals in solution due to the very acidic conditions. Then we use different acidophiles from Rio Tinto in order to grow in contact with very high concentration of metals. This was used for example, for UV attenuation studies that we published several years ago, and at the same time for running the aging studies and the fossilization studies that we were following. And we had another work done previously, in which we were able to follow the fossilization and precipitation of minerals driven by a fungi, a natural fungi from Rio Tinto. It's really interesting. These kind of environments are really interesting from different point of view, but really challenging because it's really difficult to get, for example, RNA. RNA is amazing. It's really difficult to have some results.
Any more questions, Felipe? Please ask away.
Really nice picture behind you.
No, don't you miss it, Felipe? I miss it so bad.
Yeah, absolutely. He have the advantage that has not been there.
I know.
If not, right
He
now,
doesn't have to feel the pain we feel.
plenty of water around.
Yeah.
It's in honor of you too. So
Thank you.
how about that? When we do the Antarctica one, it'll be in honor of me and JJ. And wait a minute, I take that back. And also, I think Nicole's on the line, who's been there a couple times. But okay, so I'm going to move on to the next subject matter here, and then I'm going to ask Chris if he, I don't know if Chris, you're still on. If you wouldn't mind talking a little bit about IMSA. But before we go there, one of the things that I want to do, and this is related to IMSA, and hopefully Chris will update us on IMSA, is I would like to start drawing up what I've been talking about for the past couple of years with everybody's input. As we approach these new microbiome environments, I would like to have a checklist or a document standards that we can circulate around and have a position paper possibly on the do's and don'ts of what to do when you get to a new microbiome environment. I've heard way too many stories presented to me where somebody found a new microbiome environment, a novel environment, and they just dove in and went in with like exposed skin. And I really hate to see these environments damaged by exposure to human microbiome and other elements that aren't handled properly. And I think that this group, even if it's a one-page checklist or document, can put together something that people can use in the future. Sometimes people don't even realize that they're going into an environment and affecting it. So this is one thing I'm going to start circulating this around. And if anybody has any checklist or just an idea, even if it's one thing that they can add to this, just one thing between the 80 people that we have in this group, the do's and don'ts of entering a new microbiome environment, you're going to start seeing these emails circulating around. So I'm not going to take the time to open up a discussion about it today because I want to put it into an email, more of a permanent structure. But I just want you to be familiar what email you will be seeing. And I do want everybody to participate if you have an opinion one way or another. And this was something that's born out of the IMSA group. So some of you have heard of IMSA, that Chris and I are on IMSA. And Chris, you feel like just doing a quick overview of?
Yeah, my pleasure. So good to see all the names and some faces on the call. A lot of old friends and colleagues here. So yeah, IMSA, and this is actually something that Scott and I have been chatting about, well, really, I guess since we met, is weird ways to collect samples. There's a little known fact that Scott Tighe used to purposely degrade RNA in his lab. He would just lick every Eppendorf tube he had. That's he doesn't do that anymore, but we for some older studies, Scott was in charge of degradation. So anyway, in terms of everything about the sample collection, the metadata, avoiding degradation, the IMSA it was originally the International Metagenomic and Microbiome Standards Alliance. It's now been renamed, rebranded. Krista's been helping as well. I'm glad to see Krista on the call with the website and others. And this is with NIST, so the National Institute of Standards and Technology, who has, of course, by their very nature, trying to figure out best practices, methods for benchmarking, standards, controls, everything from atomic clocks to weights and measures to biological metrics, and including human genomes and microbial genomes. So Scott and I have been working with them for a good while. We actually also have a paper coming out, hopefully in a few months, about some of the other mixtures that Scott made. And we want to, so a lot of the standards are built around having titrated controls that you can use as a positive control. When you run your samples, if you don't already, you should all have positive controls. It's just like when you run a gel, if you didn't run a ladder with your gel, how the hell would you know what size things are? You wouldn't, right? So it's the same thing here, is that if you're profiling samples, you need a positive and negative control, of course, like any good experiment. But interestingly, 5 years ago, they didn't really exist. So we've built a lot of them with BEI and ATCC and Zymo and some other groups. So we want to have it now. IMSA stands for the International Multiomics and Microbiome Standards Alliance. So it's not just looking at DNA; it's looking at DNA, RNA, proteins, epitopes, anything that you can do to a microbial sample, and use and build best practices, standards, and controls for all those. So what's exciting is it's now can reorganize and relaunch. So there's going to be quarterly meetings where we hear from industry, government, and academic groups at each one of them. And we should actually, Scott, I don't know if you sent out to this whole list, but we should send out the update once we get it from Scott Jackson.
Yeah. So I will send out the kickoff meeting. We're having quarterly webinars, and I'll send out the announcement for that if people want to join in on whether you got an iPhone or your computer or whatever. But you can put it on your Google Calendar. But it's on April 15th at 9 a.m. Pacific Standard Time or noon on Eastern Standard Time to join the meeting. So I'll send out a note.
I feel like a deja vu speaking about sampling after all the the meetings that we regularly have for perseverance. It's like a deja vu, what to sample and where to sample, and to follow the correct protocols in order to sampling to got some interesting samples. For me right now, this discussion is like a deja vu from the other meetings.
Yeah, I mean, it's the other thing we want to establish long term is not just the sampling, but is also the banking. So long term storage of these samples that can enable longer, viably frozen down cells. You can have metabolic characterization, cellular characterization. There's a lot of new spatial imaging technology. You can get really high resolution, either EM or even spatial omics mapping of some of these samples. So a lot of cool technology is also brewing that we want to build standards for. So I think, I think we actually in this group probably have the weirdest and the hardest samples. I know if it's just like something crumbing on a plate that's some version of Staphylococcus or an E. coli, whatever, right? But all these things that we're talking about here is actually going to really push the limits of the standards and the controls. But that's it. Just that it's relaunching, and we'll send that link out to everyone, and should be good meetings and good group.
Yeah, cool. Thanks, Chris. So moving right along here, we're going to close this up here in the next 10 minutes. We have multiple projects going on with XMP in the background. I don't know if anybody wants to update where their projects or their samples are, just so people are kept aware. I know that Horia has been working with JJ a little bit and Chris to get samples there. And I know that Francois has got some samples going on. I don't know if you guys want to quickly update us on your projects.
Yeah, I don't know if I see JJ on.
Yep, I'm here.
Oh, there
I'm
he
here.
is.
I was waiting for Horia to come on and talk about the samples that have arrived. But yes, we've confirmed that they are arrived and in the freezer.
Yeah, thank you very much, Scott, Chris, and Jaden. Actually, 2 weeks ago, we went on a field trip to hypersaline lakes around Transylvania. And we collected water from this hypersaline lake. We filtered the water and we extracted the DNA from the 0.22 micron filters. And we use the QIAGEN DNeasy power soil kit. And we subsampled this DNA, and one part of this DNA was sent over to Chris' lab. I think it's Whale Cornell Medicine for the nanopore sequencing. So I actually explained Scott and Chris and Jaden and Daniel that we actually aim to do this nanopore sequencing for long read sequencing, just to address a few questions on, for example, the diversity of eukaryotes or micro eukaryotes. One interesting topic for us was to see whether we have a different species of Dunaliella, and also if we have new species of anaerobic micro eukaryotes, because some of the sample were collected from the anoxic water layer in this hypersaline lake. And we also want to check with the recover assembled genome from this long-read sequencing. We want to check to validate our metagenomic assembled genome from the other subsample we prepare. So we try to make a multiple, multiple analysis on long-read sequencing, short-read sequencing, and also we do have RNA sequencing as well from the same water layers, and later on we will all compare and put it together and try to integrate all this knowledge. Thanks.
Yeah, and I think we'll plan to get them prepped and sequenced soon. I guess we just need to make sure we have flow cells ordered. But wanted to share the data with you first and the smaller team. But the degree it's possible, it goes for anyone. It's possible when I start to organize all the data and samples, and if possible, share them with people to start to have more collaborative subgroups, getting papers and some of the analyses done as a team.
And one of the things we did with Jorge's samples is he actually captured them on the columns, the DNA mini columns, did the final rinse with the DNA mini columns, spun them. He actually sent the DNA mini columns with the DNA in the actual silica straight to JJ. So we've done that a few times before, and it's worked out pretty well. And we'll have some results from JJ next time we have an XMP call to see how big the DNA is and how much DNA. And if that was a successful way for Jorge to send his DNA across the world. So
not me, the FedEx was.
It seemed to work. It was good.
Yeah.
Any other updates of projects?
Hey guys, I have to run, but I just wanted to let you guys know, you should make sure to tag me. We have a new Mars analog project that's starting this fall in Iceland. We're going to be doing some work with some groundwater systems in Iceland lakes as a Mars analog. And so I got pulled onto the team as their geobiologist. So we'll have some cool stuff to talk about soon. So I'd love to kind of introduce you guys to the project, let you know what we're going to be up to, and where it looks like we're going to have approval to actually hit the field this summer. So just want to make sure everybody knows.
Kent, I'm happy to give you some great contacts in Iceland. Some of my best friends are there, and they love to entertain.
Yeah, please do, please do. We're going to be there. We're trying to get all the field logistics figured out. This is a NASA funded proposal, so I know that they're just trying to get final approval to make sure we're actually allowed to go. Now, we found out Iceland will let us in. We just need to be allowed to go by NASA, so we're just waiting for that, and then this is probably going to happen this fall, so it would be pretty cool, late summer, early fall. So
yeah, very cool. Incidentally, we will be Robin Barbados is on the call from Krell, and I will be going up to Greenland to do some metagenomic geomapping somewhere along the dew line. So we'll keep you informed on that. We're filling out the permits right now to go up there and do that.
Well, I definitely want to talk to this group for sampling. Just I have some questions that I'd like to pose to this group as I put together my sampling plan and my analysis plan. So definitely want to talk to you guys more about it. So all right, I gotta run, guys. Take care. Great meeting today.
All right, anything else to add? Anybody in the group? Open forum, please. If not, we'll call it a day and move on, and we'll all see you again in 4 weeks. I will give you a forewarning that we will probably going to move one time around because we have a new member that we don't want to exclude. We'll keep 3:30 for most of the time, but every third time I think we're going to change it a little earlier. Because that person's at the Indian Institute of Technology and does stuff with secondary metabolite bioinformatics. So keep an eye out for that. And I think he's going to give a presentation in either next month or the month after. So there's going to be some times bouncing around here. I'll work with Krista and JJ on that to get that coordinated. So, but other than that, I have nothing else to talk about. So we will see you guys all in a month. And thank you so much for joining and attending.
Thanks, everyone. Thanks, Ken.
Okay. Thank you.
Thanks.
Great time.
Bye.
Bye.
Thank